and it's my great pleasure to introduce today's speaker, Sílvia Pujals, who is joining us from Barcelona. She is a Ramon y Cajal researcher at the Institute for Advanced Chemistry of the in Catalonia and adjunct professor at the Department of Electronics and Biomedical Engineering at the University of Barcelona. Sílvia got her PhD in organic chemistry from the University of Barcelona before moving on to a postdoc at Kyoto University, where she focused on biophysics and electron microscopy. Upon her return to Barcelona, she became a senior researcher at the Nanoscopy for Nanomedicine group and worked on super-resolution microscopy for nanomaterials. And this broad background, combining expertise in drug delivery, peptide synthesis, and optical and electron microscopy, really forms her current research aims for the rational drug design for nanomaterials for targeted drug delivery. And she'll be telling us today about correlative super-resolution and electron microscopy methods for nanomedicine. Sílvia, thank you very much for joining us, and please go ahead with your presentation.
Thank you very much, Johanna, for your kind introduction, and all the people of Euro-BioImaging for this very nice organization. And I am also very grateful to COMULIS Cost Action, because they are very nice also in embedding me in the community. As Johanna said, I'm a chemist. Mostly people working in correlative imaging or in imaging are biologists or physicists, and I am the black sheep there, no, being the chemist doing strange things with nanoparticles, but they have been very nice, no? Even they invite me to give talks in the network, so I am really grateful for it. I hope you can see my screen well now. Okay, so then I will, thank you. I will start with my presentation. So as Johanna said, we are focused on nanomedicine. So nanomedicine means applying nanotechnology to medicine. There are different fields within nanomedicine. We focus on drug delivery. So nanocarriers that are used for different purposes like imaging and diagnosis, disease prevention, no, the vaccines, especially last years with COVID, we know that nanoparticles are really useful for this topic, and we focus on drug delivery. In the case of drug delivery, compared with a classic delivery of small molecular weight drugs where there's no selectivity towards the target, with nanoparticles, we expect that they accumulate specifically at the target site. This is the theory. In fact, nanomedicine has been controversial the last years. We have been facing some problems that mainly come from toxicity and safety evaluation, the difficulty in scaling up the production of nanoparticles, the lack of models for disease, and the poor targetability. In this article here, they showed how only 0.7% of the nanoparticles administered in the body reach the final target. So to face these problems, what we do is to use advanced microscopy techniques to tackle the different aspects of the life of a nanomedicine, from the formulation and characterization, the circulation in blood, the extravasation and diffusion, cellular targeting, and intracellular localization and activity. If we think about the formulation and characterization of nanomaterials, there are different aspects, like there are different materials, different sizes, surface. Here it's very important the ligands. You have most of the nanoparticles for drug delivery. They have a targeting ligand that has to be well exposed in order to get the specific targeting to the specific cells that you aim for, different shapes, and so on. So these different properties, physicochemical properties, have a very high impact in the biological performance. Only small changes in it will change all the route of administration and the performance of these nanoparticles. And it's very important to characterize them before applying them in a biological media. So this is very important, because if low molecular weight drugs, a copy of them, it's the same, like if you have doxorubicin, for instance, that it's an anti-cancer drug, all the doxorubicins in your spot will be the same. But in the case of nanoparticles, we face very high heterogeneity. So for instance, they can have different sizes, different ligands, number of ligands on the surface, and so on for the different properties. But the techniques that are mostly used are ensemble methods, the methods that are used for conventional drugs. So then what we get is average values that most of the times are not representative from the population of nanoparticles that we have on our sample. That's why we think that we have to move to single particle methods in which you can see all the heterogeneity, you can learn about the consequences of nanoparticle performance, and you can work on learning about the interplay between different parameters. That's why we propose to use advanced microscopy methods, correlative methods, and that include super resolution and electron microscopy. So at the beginning of the talk, I will focus on the only super resolution methods, and then we will move slowly to the correlative methods. So why super resolution? You know that in conventional microscopy, there's a diffraction limit set by Abbe law, that it's around 250 nanometers. So nanoparticles that are usually around 100 nanometers cannot be seen at the single particle level with the conventional microscopy. So then different families of super resolution microscopies have arisen in the last years. There are three main families, like SIM, STED, and single molecule methods. We will focus in single molecule methods. SIM and STED are also really nice methods, and I think they are great, mostly if you have live samples to image. But we focus on single molecule localization because it gives the greatest resolution, despite it takes the longest time. So if you want to work with cells, then you need to fix the cells, because as you will see, we have to acquire a lot of images to get the super resolved image. So then it's tricky to work with live samples. So how does it work? So if you have your structure of interest labeled with fluorophores, in a conventional image, all of the fluorophores will shine at the same time. So then you will just see a very blurred image. The trick of super resolution microscopy, of single molecule localization microscopy, in this case is a STORM. Many acronyms are used depending on the specific technique. In a STORM, what you do is you use a trick to have the fluorophores in a photoswitchable way, that most of them are in the off state and only few of them are shining. Because only few of them are shining, around 1% of the total, then you are sure that you can fit a Gaussian on the emitting signal and then have a localized data point. So you do this typically for many, many frames, 20,000 frames, 50,000 frames. And at the end, that's why I was telling you that it's very long acquisition. It takes minutes to get all these images. So at the end, you add up all these points and get the reconstructed image. Okay, so it works like this. So it's like the blinking of the Eiffel Tower. Each of these blinks you detect and you add, and then you add these points at the end in the reconstructed image. So you need fluorophores in the places you want to image to get the final structure at the end. Like here the antenna that it's not labeled, you will not see it at the end. So in reality, this works because we use a very special buffer, the STORM buffer, that has some reducing agents, some thiols that intercalate in the fluorescent dyes that have to be specific dyes. And then because of this intercalation, most of the signal is in the dark state and only few of them are emitting. Another way to have single molecule localization microscopy is the paint methods, that instead of relying on the photo switching of dyes and this special buffer that we use, what they do is to label the structure of interest with an oligonucleotide. And then in the supernatant, you have the complementary one with a dye. Then every time that the complementary one binds to the structure of interest, then you have a blink in the microscope. So you are following these binding and unbinding events. And this is at the end what you will use to reconstruct your image. Okay. Because it relies on these oligonucleotides, and you can work with many different sequences. In theory, well, many of them. Then with just one color of the imager, you can have many structures of interest labeled in your sample. No? So each time, like here, you use microfluidics. No? You have first imagine the imager 1, so that it binds to the structure that has the oligo complementary to imager 1. Then you wash at the imager 2 that intercalates with the one that has imager 2 complementary sequence, and so on. You can do this n times. And then only using the same fluorophore, you can image many colors. Like in this example here with DNA origami, when they can image like 10 different structures with the same fluorophore. So it has very high multiplexing capability. And this is the same, but with cells. So they image microtubules, mitochondria, peroxisomes, and so on with this method of DNA paint. Okay, so there are these two modalities in single molecule localization microscopy. So this has been applied in biology. You can see here very nice examples of mitochondria, nuclear pores, and so on. But very few people has applied to chemical structures. So we were one of the first to apply it to chemical samples. And this is one of the first examples. It's a quite standard molecule that is diphenylalanine, so only 2 amino acids that in water they self-assemble in these very nice fibers that you will see. And what we did is this paint approach. No, we had not a oligonucleotide in this case, but the self-assembled motif, FF, labeled with a fluorophore, and then each time that this fluorophore was binding through FF to the structure of interest, then we got a localization. And we did this for many frames. This is what we saw in the microscope. No? Each of these points is a binding event to our structure of interest, and then we could reconstruct the structure. So we could see this type of images, and we could learn about the self-assembly process of this molecule. We moved to more challenging structures, like in 3D hydrogels, in this case FMOC, FF, so we could see in 3D how this is behaving. We could learn about the mesh size, and here are other examples, like for instance micromotors, in which these are, in this case, big nanoparticles, no, that have an enzyme on the surface, and then they can move. And with super resolution, we could learn the distribution and the number of these urease enzymes on the surface of these nanoparticles. Another example is the polyplexes for gene delivery. So we could learn about polyplexes are a mixture, no, of polymers and DNA or RNA, and then normally they are mixed at different ratios, and we could learn by using a storm and labeling each of the components with a different color, no, the amount of each of these components at different ratios. So until now, no, what we have seen is the structure or counting of molecules in different types of structures, but we wanted to go further, and we wanted to quantify the the functional ligands, because this is a key concept in nanomedicine. In nanomedicine, as I told you, we have most of the times the nanoparticles decorated with some type of ligand. In this case, it's an antibody. So this antibody binds to specific receptors that are overexpressed in cancer cells. And this ligand biotarget recognition, it's very important for having a more specific target of the nanoparticles. So we want to learn how many of these are on the nanoparticles, how are they distributed, and for that we use DNA paint. So we had our nanoparticles with the antibodies, then we added the ligand to these antibodies with this docking strand, this oligonucleotide that will allow us to perform DNA paint later on. And as you can see, only the antibodies that are well oriented could bind to the ligand. So only the functional molecules, the antibodies that later on will interact with the receptor will be counted with this technique. Okay, then we flow on the complementary strand with the fluorophore, and in this way we could analyze and quantify the functional antibodies on the nanoparticle. This is just a scheme. The reality looks like this. This is the nanoparticle, and each of these points is a binding event of the oligonucleotide on the surface of the nanoparticle. As you can see here, it's just two examples, but it's quite heterogeneous. We can have this type of histograms, and some nanoparticles have more, some have less. And also, as I told you, this technique allows for a very multiplexing. You can have different antibodies on the surface of the nanoparticle, and then flowing the complementary sequence for each of them, you can quantify, like in this case, 3 different types of antibodies on the surface of the nanoparticle. Here it's another example with DNA paint. In this case, so here they were synthetic nanoparticles. In this case, it's virus-like particles that are nanoparticles that are made by cells. So this comes from the influenza virus. So you transfect only the proteins that allow the self-assembly of the viral structures. So there is no DNA content or RNA content at the end. They are just used, the aim is to use them for drug delivery, so they are not harmful. So what you do is transfect these proteins on cells, and then they produce virus-like particles. Okay? So what we did is to quantify the amount of each of these proteins on the surface of the cell or on these virus-like particles by DNA paint. In this case, we did immunostaining with the primary antibody and the secondary antibody having the complementary sequence to perform DNA paint. So in this way, we could count and see the distribution of each of these proteins on the surface of cells, that this part I will not explain, but also on the surface of these VLPs. So this is how it looks now in the microscope. You can have the VLP and then each of the proteins, and you can quantify. And then here it's nice because you can go VLP, so at the single particle level, and correlate the amount of one protein and the other, and have this type of graphs that allow to see if there is a correlation between the number of one protein and the other. And this we did for the 3 proteins that were expressed in order to produce the VLPs. So HA, NA, and M2. Okay. Now I am going to enter into the correlative methods that we have. So until now I have shown you how we can use DNA paint or other single molecule localization techniques to have multicolor imaging, nanometer resolution, and we can quantify molecules. In this case, the ligands on the surface of a nanoparticle. but we wanted to combine this with electron microscopy, because then in electron microscopy, we can have a direct visualization of the structures and have morphological and size information. So we join both to get this CLEM method. I will explain you how we do it, but to get information on a multiparametric level at the single particle level and single molecule level. So how we do it? So we formulate the nanoparticles. These are polymeric nanoparticles. And then in this case, our ligand here will be the oligonucleotide sequence for performing DNA paint. So we have nanoparticles with different formulation, with different percentages of this oligonucleotide. And then we attach them on a TEM grid. We prepare the chamber with the imager that will allow us for the imaging of the ligands. And then, well, first we need to know, because later we will do correlative. We need to know in which of the grids we are imaging. So this is important to acquire some bright field images and to learn in which grid hole you are. Then you do all the DNA paint imaging to get information on the number of ligands on the surface of these nanoparticles. Then you take the grid out of the chamber, you do negative staining, and then you go to the TEM to do TEM imaging. And then later on you stitch all these images and perform the correlation. So yeah, this is the work of Theodora. Most of the things that I will present on correlative has been done by Theodora, PhD student that did, she had only 3 years to do the PhD, and she did a titanic work on in vitro, as I am showing now, and then later on cells too. So yeah, this is the type of images that we obtain with DNA paint. So each of these are nanoparticles. In red, you have the number of ligands on them. And then this is the TEM image. And then we get the correlative images. Okay, so this, to our knowledge, is the first super resolution electron microscopy correlative method for nanomaterial characterization in vitro. And it's not only beautiful images, and as you can see here, but we can get a lot of data out of it. For instance, we can learn, as I was telling you, about the number of ligands that we have on the surface of the nanoparticle. So we can formulate different formulations with less or more number of ligands on the surface and learn about by counting and applying Q-paint, the number of ligands that we have on them. Okay, and this was surprisingly low. It's much less than what is expected in theory. Okay, so this method was really good for us because we could learn, and then this is a technicality, but it's really important. So seeing the results, what we could do is go back to our formulation and change it in order to have more ligands exposed on the surface of the nanoparticle. So if this was the formulation that I showed you before, what we did is to change it and have a short peg. I will explain you now. So here there are brushes on the surface of the nanoparticles, and some of these brushes have ligand and some of them not, because you have certain balance of ligands on certain density of ligands on the surface of the nanoparticles. So what we did is to shorten the brushes that have no ligand. As you can see here, the ones that have no ligand were shortened in this new formulation, and in this way the ligand was more exposed, and then we could get higher number of ligands. Okay, so you can see the huge difference. For instance, in 10%, in this formulation, we get like less, I think it was around 5 or so, but with this new formulation, we get around 40, 50. Okay, so microscopy, I know it's very technical this, but it's really important for me because with what we learn with microscopy, we could go back to the formulation and improve it. Okay. So yeah, this is and this is another type of representation. So we have different percentages, and we can do is do this multiparametric analysis. No, so correlate the number of ligands with the diameter and get this type of graphs that then we can filter, like for instance here, no, and we can know that the smaller nanoparticles have less ligands than the bigger ones. Okay, that this is what we were expecting it, but to see it in reality, it's really nice. And these were really unexpected. And this data here, you cannot learn if you don't do the correlative method. Okay, what we observe is these 0 ligands here or 0 ligands here, that a lot of nanoparticles, no, that we could see by TEM had no ligands in DNA paint. Okay, so if you do only fluorescence imaging, all of these will be underestimated because you will not be able to see them. So around 30–40% of the particles have no ligand functionalization. So it means no receptor targeting. So this is quite dramatic. So with this data, I think we have to go back to the formulation and improve it. Okay, so we with this method, no, we think that it allows for a smart optimization of the nanoformulation at the single particle level. And now what we want to go is to see what is the repercussion on performance. Okay, so go to the biological questions here, no, take all these nanoparticles and see how they perform, no, with these properties that we can learn about at the single particle level. Yeah, this is just another example, now that we are in vitro, that is with this type of fibers that are supramolecular polymers. So not only like a spherical standard shape nanoparticles can be used, but more sophisticated materials can be also observed, no, with this method here, as you can see. Okay, so yeah, moving to the more biological part of the talk. First, I want to explain about the intracellular localization and activity here. So if until now we have talked mostly about the physicochemical properties of the nanoparticles, now I want to talk about different aspects. The first one is about the intracellular trafficking of these nanoparticles. This is very important, no, for drug delivery, because there are many articles claiming about how the drugs escape the endosomes, if it's degradation, if it's endosomal escape, but there are not many techniques that allow to visualize this. Okay, so we are working in them. So yeah, with light microscopy, you can have a spatiotemporal localization of the molecules. It's very specific and you have high sensitivity. It allows for multicolor imaging. But then, yeah, the resolution limit is there, no, and you have no cellular context. So that's why electron microscopy comes in play. No, you have sub-nanometer resolution. The ultrastructure is excellent. You have direct visualization, and you don't need a specific markers, but it's incompatible with live wet samples, have a limited molecular specificity and no temporal information. Okay, that's why correlative methods come into play. And then, yeah, you can correlate the same region in both imaging modalities. You combine advantages of both techniques and high resolution analysis of cellular events in the cellular context. Okay, but these techniques are not yet optimized for the study of nanomaterials. Okay, the first example that I will talk about is this method that we developed a few years ago that is for bacteria, but I think it could be easily applied to materials. So in this case, it was the study of the intracellular trafficking of Salmonella. So to label it with fluorophores for storm, what they did, the collaborators in Leiden University, is they start the Salmonella with analog of methionine, homopropargyl glycine, that has this handle here. So then the proteins in Salmonella are expressed with this amino acid. And then later on, what you can do is to click with click chemistry, very specific reaction, very bioorthogonal, so there is no background in the labeling, with a fluorophore specific for storm. And then you can perform both storm and then electron microscopy to have correlative images. So the protocol is like this. So in this case, we did cryosectioning of the dendritic cells incubated with Salmonella. Then in the section, we did the click chemistry reaction. We also labeled the nucleus to have some signature signal, that it's easier to follow in low resolution. Then we mounted the sample with the Glog's buffer to perform super resolution microscopy. So in this case, also again, it's important the reference here. So we knew where in the grid we were imaging. So we did first the storm imaging, then we take the grid out, we stain it for electron microscopy, and we go to the electron microscope. Again, we find the same area as before, and then we can do the time acquisition, and later on we do the correlation of both techniques. Okay, so this is a low resolution image, and this is just the low res. Here you can see how the resolution is improved by using storm. And here is adding the ultrastructure that electron microscopy gives you. And if you zoom in, what is interesting in this case is that, and you cannot see it by low resolution, is all these like small pieces here that the people working with bacteria told that they were secretion proteins. Okay, so this allows for it's a nice method to study the intracellular trafficking of bacteria. Then another example, in this case with nanomaterials, that is the one that we are mostly interested in. This is the latest work that we are doing. Theodora is finishing the PhD, and I will present her latest data. So when working with cells and wanting to do correlative methods, there are mainly two ways, either the standard room temperature methods with the standard resins, or the cryo methods. And then in between, there is this free substitution, in which, well, half of the protocol you do in cryo conditions, and then you move to the room temperature methods. Okay, so we discarded the totally cryo method because for us it was too complicated, but we wanted to compare between the standard techniques and the free substitution. So I can skip. So we tried epoxy resin, LR white resin, and then the acrylic resin with high pressure freezing and free substitution. And analyzing the ultrastructure, we saw how with free substitution was for us the best method, and also, despite being the longest protocol, because these two are much faster, we choose that one also because not only the ultrastructure was greater, but also the fluorescence could be maintained, because in these two, mostly osmium is used, so it kills quite a lot of fluorescence. So this is the protocol that Theodora has been using. So we incubate the cells with the nanoparticles. We do a mild fixation. We get the pellet, no? Then we do high pressure freezing, freeze substitution, then the sectioning. We add the fiducials, the store buffer. Then we do the storm acquisition. And then we retrieve the grid from the chamber. We do the staining and then the TEM imaging, the TEM stitching, and then using IC software, we get the correlation. So as I told you, it's very nice ultrastructure, as you can see here. The fluorescence is very well preserved. So this is the channel of the nanoparticles. So these are the same nanoparticles that we did the in vitro correlative method before, no? So these polymeric nanoparticles. And then the fiducial marker to be able to do the correlation of both images later on. We are using IC software, that it's kind of semi-automatic software to get the correlative images, and then we get this type of images. In this case, it's with low resolution. This is for 4 hours. So what we observe now is that the signal is located in different types of organelles. So sorry, I'm moving in different types of organelles, like late endosomes here, or early endosomes here or here. And when we overlap it with the super resolution image, with the storm image, we can see how they nicely fit and they can be correlated very well. No? So compared with the low resolution signal, storm is more quantitative and it fits better the resolution of electron microscopy. So we did this for different timings. This is in low res. You can see again, no, how in low res we cannot locate very well the nanoparticles, but here in a storm we can see the individual particles, and we can assign in which compartment they are, the number of them, if they are intact or degraded. This we are working on. And then we can assign, no? For each of the images we obtain, we can see, this is a lysosome, this is a late endosome, and so on. No? This is early endosomes. And this Theodora has done for many images, for many time points, in order to be quantitative. Okay, so we can see, no, if they are outside, if they are in early endosomes, in late endosomes, in lysosomes, or this part we are still working in, what happens after, no? If they are degraded, or if they are expulsed out of the cell, or what's going on. Okay, but for the moment we have done up to 24 hours with these polymeric nanoparticles, and for each time point we have evaluated how many of them they are in each of these steps, so we can get this type of information. No? So what's happening in the cytoplasm, in the lysosome, they are increasing, late endosomes and early endosomes in the earlier time points, and then they go down. Okay, so we can be quantitative thanks to these correlative methods. And we have applied a drug, a drug that it's quite famous, chloroquine, that what it does is neutralizes the trafficking of endosomes, so then they cannot mature into lysosomes. And again, we can quantify and see how without chloroquine the endosomes evolve in time to late endosomes and lysosomes, but with chloroquine we have more of the early endosomes. Okay, I don't know if I still have time, because I want to talk briefly about the last application.
Yeah, there's still time.
Okay, then I will talk about another topic that is important in nanomedicine, that is the extravasation and diffusion. No? So the nanomedicines mostly are in the bloodstream, no? And then they have to cross the endothelial barrier to reach, well, for cancer, a tumor, no? Or the inflammatory site. So for this, I will explain you that we use organ on a chip, okay, as a model. And then I will show you the imaging that we have done for it, that for the moment is separate. So we have done optical microscopy and then separately electron microscopy. Okay, not yet correlative, but I think, yeah, it's a nice research to explain here too. So as you know, most of the preclinical study methods are done in 2D cell cultures or in animal models that are, the animal models are in 3D, but they have a different types of cells. No, they are not human cells. They don't show the same phenotypes. So that's why organ on a chip is a nice alternative, no, at least in between 2D and animal models, no, to have something in between. So here you have the same cells as in the 2D, so you can work with human cells or human-derived cells, but you can have a 3D architecture. There are many examples of many different organs. And in our case, now, because we are interested in nanomedicines against cancer, I will show you our humble contribution in cancer on a chip. But you can check, and there are many applications now. There's this lung on a chip, gut on a chip, heart on a chip, and so on. So what we did is we work with this commercially available chip that has a central channel and a lateral channel. And in the central channel, we have the extracellular matrix, collagen in this case, and a spheroid made of cancer cells. Okay, so you can see here the spheroid. And then in the lateral channel, we work with endothelial cells, IBEC, forming a barrier, an endothelial barrier. And then what I will explain you today is how we flow nanoparticles here, and then we learn if they cross and reach the tumor, the spheroid, no, in this case, or not. Okay. So we had two models, the healthy one and the cancer one. So in the healthy one, in the central channel, there are no cancer cells. Okay, so in this way, the endothelial cells are very healthy and they form these zips. Okay, so these are called tight junctions. So they are very strong bindings between the different cells here, so that most of the molecules cannot cross this barrier. Okay, you can see here the 3D reconstruction. This is in confocal, no super resolution here, but you can see how the barrier is very well formed. And then when we flow nanoparticles here, so they are not able to extravasate to the central channel. Okay. Then what we did is to work with the cancer model. So in this case, it's the same, but in the central channel, we have these spheroids made of cancer cells. And then because these cancer cells release some chemicals, some cytokines and so on, what happens is that the endothelial barrier is not so well formed. No? You see all the zips that we saw before, like you can see here, no? In the healthy model, all these zips. In this case, they are kind of destroyed, no? So there are many holes. And when we flow the nanoparticles, they can fastly extravasate into the cancer cells. Okay. And then, because these micelles have very nice feature that depending if they are assembled or not, we can do spectral imaging, so follow at different wavelengths and learn if the micelle is still like a micelle or if it has been destroyed. No? So this is in 2D, in which in the medium mostly it's a micelle, but then in the cell membrane it seems that it disassembles, no, and it's mostly a monomer. and also inside the cell. Or some of them get endocytosed and then they are still in the micellar form, but some of them disassemble in vesicles. Okay, so we use this spectral imaging in the chip. Okay, so we could follow in space, no, in the different channels, in the flow channel, in the barrier, in the extracellular matrix, and in the cells, in time, what was going on with these micelles. Okay, so you see how in time, no, they are able to extravasate and reach the tumor cells. Okay, so yeah, this is maintained under flow, and then in contact with the cuvette, it seems that they disassemble, and then they get closer to tumor cells. So this is so far what we have done with organ on a chip with optical microscopy. And then we wanted to also do some electron microscopy. No, it's, as I told you, these tight junctions are very important, no, for the extravasation. And then we went to a simpler model, this blood vessel. So there is this serpentine chip in which here we see these endothelial cells, no, this cuvette, and then they align and form the barrier. No, you can see here in 3D, and we develop a method to cut this chip and be able to see the ultrastructure with electron microscopy. So what we did is we did again free substitution, and then we had the polymerized chip. Here the problem is that the chips are made with a plastic that is very soft. So then when cutting directly for ultra thin sections, it doesn't work. So what we had to do is to cut out most of this material, no, of this plastic, and then re-embed in resin, no, to be able to cut with the ultramicroton. And then with this, we went to the electron microscope, and we could see these tight junctions, no, that are in this, that are forming this blood vessel on a chip. Okay, so this is the last example I wanted to give. So to sum up, I have explained different super resolution methods, CLEM methods for nanomedicine. So we can go from the very structural information at high resolution to be more quantitative and learn about, for instance, ligand number, do correlative methods to have multiparametric analysis. No? In this case, I've shown you ligand versus size or shape. And then we learn detailed information on the performance, no? On the trafficking, endosomal escape, and so on. Mostly I presented in 2D, but we are going into 3D with this organ on a chip example that I gave you. So what we want, no, to do is to link these physicochemical properties of the nanomaterials with their performance in order to go towards a rational design of nanomedicines. And there are many, many future prospects, so we can expand to other materials, other types of microscopies, to learn about which nanomedicines are the ones that are working. So going to screening, personalized medicine, and so on. So if you want to learn more, we have some reviews about the topic. And I want to thank, so the group that I was now, the group that I am now, the collaborators, and yeah, I want to thank my former professor, John Hoyler, that he teach me everything I know about electron microscopy, and we spend many times in the microscope, and he teach me a lot. And yeah, especially Theodora, that she has done a very nice work both in vitro and in cells and in a very short time. Okay, and again, thanking Euro-BioImaging for organizing, and COMULIS also. For the people that is not part of COMULIS network, I encourage you to check the web and all the actions they have. They have meetings, they have fellowships, for the students to go and learn about the different methods, training schools. So it's a very nice network. And thank you very much for all your attention.